Thank you so much for joining us today for the ATS Journal Club, sponsored by this section of Genetics and Genomics. My name is Rob Mettelman. I'm a postdoc with Paul Thomas's group at St. Jude Children's Research Hospital. And today we are really excited to discuss a recent article by Markov et al. Entitled Distinctive Evolution of Alveolar T-cell Responses is associated with clinical outcomes in unvaccinated patients with SARS-CoV-2 pneumonia. And this study was recently published in Nature Immunology this past August. As a note for all the folks online, this journal club is being recorded and will be posted on the ATS website for later access. Now, presenting the article today, we are honored to have the co-senior and corresponding author of this study, Dr. Luisa Morales-Nebreda, from Northwestern University Feinberg School of Medicine in Chicago, Illinois. So welcome. We are also joined by three panelists: Dr. Sophie Valkenburg from the Peter Doherty Institute for Infection and Immunity at the University of Melbourne. We have Dr. John Alcorn, who's in the Department of Pediatrics at Children's Hospital of Pittsburgh, and Dr. Jie Sun, who is in the Department of Microbiology, Immunology, and Cancer Biology at the University of Virginia. So welcome all. A little bit about today. So we'll start with our journal club presentation by Dr. Morales-Nebreda, and then we'll move on to a question and answer / discussion section with the panelists. So if you're joining online and you would like to pose any questions, as they come up, please type them into the chat feature of the webinar, and then during the panel discussion, we will try to get to as many as we can. So without further ado, I'd like to introduce Dr. Morales-Nebreda to share a little bit about her work with us. Thanks.
Thanks, Rob, and thanks, Katie, for the kind invitation to present part of our work. Truly an honor to be here. Also, thanks to Sophie, John, and Jie for joining us, expert discussants. I'm really looking forward to your input and insights and feedback on this work. So I don't think I need to really tell this crowd, but we know that pneumonia is really the most important or the leading cause of death from an infectious disease process. And this not only includes SARS-CoV-2, but also includes other types of virus-induced pneumonia like influenza and bacterial pneumonia. So I think in this current climate, I think although this is obvious, I think it's important to remind us that we're studying really something that is very important for public health. And so part of my work in my lab is really focused in trying to understand what are the mechanisms that drive the development of and the recovery from acute lung injury. And so as part of virus-induced lung injury, we know that it really manifests clinically as the acute respiratory distress syndrome. This is a syndrome that is typified by an exuberant inflammatory response that floods the alveoli and really ends up disrupting the alveolar epithelial capillary barrier. And this leads to impaired gas exchange and respiratory failure. So in my lab, we really try to focus on understanding how different resident immune cell types change over the course of pneumonia and drive the repair of this damaged barrier and the recovery from this type of pneumonia. And so really what we try to also focus on and what would be the primarily the topic that we'll be presenting today is how different lung T-cell responses are actually invoking protective or pathological immune responses in the lung. And so to do this, we really leverage a system biology approach center that was created at Northwestern back in 2018. This was under the leadership of Rich Wondering, at that time our MICU director, where we actually posited that if we were able to understand the mechanisms of host and pathogen interactions within the alveolar microenvironment throughout the course of pneumonia, perhaps would be able to understand better the molecular correlates of protection from pneumonia. And so in SCRIPT, what we do clinically is that we take samples from patients in the intensive care unit who have been mechanically ventilated or intubated for respiratory failure. These are clinically indicated BAL, so it's part of our clinical work. When a physician sees a patient and suspects they might have pneumonia, a BAL is indicated for this diagnosis. And so those samples are sort of split, and part of it goes into our laboratory course where flow cytometry, and we sort this immune cell subsets, and we have a big group focus on macrophage biology and T cell biology. And so these samples then undergo downstream analysis from multiomics technologies. And then I think what's really we try to leverage is understanding those molecular signatures in the context of a clinical scenario. So understanding what's going on to the patient at that time from a clinical perspective to actually gain insights into the biology from our samples. And so back in 2021, we had been collecting the samples, for 2-3 years prior to this. And one of the things we really noticed back then is that really the severity of illness and mortality was not that different in patients with different types of pneumonia. So to walk you through this is non-pneumonia controls here in blue or light blue, which are patients intubated for respiratory failure for causes that are not related to pathogens. COVID-19 is COVID-19. Other viral pneumonias are pneumonias that are caused by non-SARS-CoV-2 viruses, and other pneumonias are bacterial, mostly comprised by bacterial pneumonias. So what we saw initially during the pandemic was that these patients had similar severity of illness, demonstrated here by a SOFA score, and their mortality was not significantly different, although it's slightly less for SARS-CoV-2 patients in this cohort. But the one thing, most of us who were at the bedside during the pandemic and treating these patients, one thing we noticed that was really the inspiration for this work was how prolonged the clinical course of these patients was. So you can see here to the left that the length of stay of these patients in the ICU and the duration of their mechanical ventilation was substantially and significantly increased for patients with SARS-CoV-2 pneumonia when we compare that to patients with other types of pneumonia. And so this really prompted us to think about what were those mechanisms that were driving that sort of protracted clinical course in patients with SARS-CoV-2. And so from this, and I won't go into much detail, but it's sort of the preface to what the T cell story was, is that during this time we postulated a model for SARS-CoV-2 inflammation or SARS-CoV-2 alveolitis. And this is really inferred by known biology and also by the findings that we had with this research. And so this is a regular, known alveoli, which has a tissue-resident alveolar macrophage, a type 2 cell that expresses ACE2, and then we have sort of the preponderance of AT1 cells. What we found with this model was that perhaps SARS-CoV-2 enters the distal alveolar space, infects AT2 cells. Perhaps it also infects tissue-resident alveolar macrophages that send these T cell chemokines that in turn attract T cells into the alveolar space. These T cells produce interferon gamma, which then leads to death of that tissue-resident pool of macrophages that is the most preponderant immune cell in the alveolar space. That then leads to the replenishment of that pool by monocyte-derived alveolar macrophages, which are then in turn secreting more chemokines and cytokines that continue to attract T cells. And this forms sort of a positive feedback loop between these two resident immune cells that drives a persistent inflammatory phenotype in these patients. And so based on this, we're really interested in asking the question that SARS-CoV-2 might be clinically different from other types of pneumonia. And so we really wanted to understand, are there T-cell specific phenotypes or molecular signatures that can actually explain part of that difference that we see at the bedside and can give us insights into more of the molecular mechanisms that are driving these distinct phenotypes. And so I'll walk you briefly through the anatomy of this script cohort. So we perform both flow cytometry, bulk RNA sequencing, and TCR sequencing, and I will go through this sequentially. But the first part was analysis of flow cytometry. We have a cohort of 270 plus patients having more than 400 samples, and some of these samples were actually longitudinal samples to try to give us a better idea of the temporal changes through T-cell responses. And I think something that is interesting about this or unique about this cohort is that these were really early pandemic samples. So the samples were collected between 2018 and the first wave of SARS-CoV-2, so up until July of 2020. And these were all unvaccinated patients, right? And we are analyzing here CD8, CD4, and regulatory T-cell samples. And so the first observation that we had, and I won't go too much into detail here, but just to sort of recapitulate the sort of the anatomy of this cohort, were patients, the majority of them were, most of them were male patients. The median age was around 62. The ethnicity was pretty well balanced, except for SARS-CoV-2. Most of our patients were Hispanic population. And again, we see, I guess most importantly, we see a similar distribution of severity of illness among our patients. This unfortunately came up a little blurry. But the first thing we did was that we performed hierarchical cluster on the abundance of immune cell subsets in the alveolar space. And I'll walk you through this. The diagnosis here in blue is non-pneumonia control. Yellow is other pneumonia. Red is SARS-CoV-2, and green is other viral pneumonia. And you can see here, even though it's blurry, that the majority of SARS-CoV-2 samples are actually clustering with an enrichment for T-cell subsets. Now in yellow here at the top, right at the column, you see most of these samples have actually a neutrophilic enrichment, which is sort of known biology about pneumonia is a neutrophilic alveolitis or neutrophilic inflammation. And then we see that macrophages were enriching samples that were mostly seen in non-pneumonia controls, which also aligns with known biology. So having seen that this samples were actually enriched for T cells in the alveolar space, the next thing we did is we asked whether the samples, we divided them into samples that were early or late, so samples that had been collected before 48 hours and after 48 hours from initiation of mechanical ventilation. And the thought behind this is that most infections that occur within the first 48 hours are likely the result of infections that occur prior to the initiation of mechanical ventilation. So this was the reason to separate them into early and late. And then infections that occur after 2 days of being intubated might have been infections that were acquired in the hospital. So this is sort of a clinical known association, and this is our decision to divide the samples. And again here, what you can see, and this is CD3 positive cells, so CD3 T cells. And so what you can see again is that SARS-CoV-2 shows an enrichment for T cells in the early stages of severe disease. I think this is important to mention. This is severe pneumonia. Now we see that this sort of enrichment for T cells actually declines in time, right? So this is time from initiation of mechanical ventilation. And what you can see is that actually this enrichment for T cells sort of downtrains throughout the course of disease. Now, the next thing we saw was that, let's see if this, give me just 1 second to see if this shows better. Oh, no way. All right. Okay. Just trying to figure out what happened here. But anyway, the next logical question was, if we're seeing enrichment of T cells in the alveolar space of patients with severe SARS-CoV-2 pneumonia, is that enrichment associated with clinical outcomes? And indeed, what we found was that patients that had an enrichment here in blue were those patients that were actually successfully discharged from the hospital. The patients in orange, unfortunately, were those patients that succumb to SARS-CoV-2 pneumonia. And so what you can see here is that there's a significant association between having T cell enrichment in your lungs and being discharged from the hospital. Now, what was interesting is that that association was relative to time. So if your enrichment of T cells was persistent, as you can see here in light blue, then you were able to probably had more chances to be within the sort of the discharge bucket of patients, right? But if your T cells actually decrease, which is what happened to the majority of samples here from patients that actually had lower levels of T cells, then those patients were more likely to succumb from SARS-CoV-2. Now, the next sort of interesting question was that we observed that this T cell enrichment was positively associated with having a good outcome. And so the next thing we asked was whether, and this is only looking at SARS-CoV-2 samples, is whether what was actually of those, because there are many things that we measure in the ICU in patients, right? We're taking laboratory samples every day, doing biochemical assays on blood from these patients. We are monitoring ventilatory measurements in them, and then the ICU is a data-rich environment, so we have a lot of sort of clinical input that is checked on a regular basis. So we try to put some of those variables here at the bottom that are regularly checked in an ICU setting, and then correspond that or correlate that, this is Spearman correlation, with the different immune cell subsets in the alveolar space. And so what you can see here, I'll walk you through. So at the top row, which are neutrophils, is actually positively correlated here in sort of darker orange with a lot of things that are not good. So being longer on the ventilator here, having more degree of superinfection, sort of added bacterial infections in a patient with SARS-CoV-2, having a higher SOFA score, having a disease status, a higher driving pressure. This is a ventilatory measurement of how non-compliant your lungs are. And then conversely, right, what we see in the different T cell subsets is that those measurements are actually anti-correlated. So here they're in blue. Now, interestingly, one of the things that was positively correlated with having increased T cell subsets in your lungs was respiratory system compliance. So the higher you have compliant lungs, the more healthy they are, within a range. Most of patients with our ARDS actually have low compliance. So this was really reassuring that this enrichment for T cells was actually correlating with important clinical measurements that we know are physiologic readouts in the ICU setting. And so we next, having really observed that these T cells were correlating with clinical outcomes, we then, I guess the next question was, are there molecular signatures that these T cells are expressing that could actually give us some mechanistic insights into what is driving their protective phenotype? And so for this, we perform bulk RNA sequencing. We did this in samples that had sufficient high-quality RNA. And the first thing we observed is that when we did K-means clustering, we found close to 1,000 differentially expressed genes. So what you can see here, every column is a sample. The sort of the rows are color-coded by diagnosis, outcome, as you have seen before, infection status, and then days from intubation. So the darker the green, the earlier in the course, the lighter the green, the later in the course of disease. So what you can see here is that we were able to find two clusters, and the cluster number one was actually dominated by SARS-CoV-2 samples. Now, when we looked at the genes that were differentially expressed in this cluster, some of and also gene ontology programs that we saw upregulated in this cluster were those related to cell proliferation, activation, monocyte and T cell migration, tissue residency, and then some inhibitory molecules as well. Now, most of the other non-SARS-CoV-2 samples were actually clustering together in cluster number 2. And so then the next thing we wanted to explore, again, was how are these differentially expressed genes actually correlating with different gene programs or hallmark programs. So we performed gene set enrichment analysis with a specific clinical variables, as you saw in the flow cytometry plots, where we actually took these differentially expressed genes, did correlation with gene set enrichment analysis, and then leading edge analysis, which actually looks into what are the genes that are actually driving a given program. And so what you see here is that in diseased patients, the top upregulated program in patients that succumb to SARS-CoV-2 again was TNF signaling via NF-kappa B. Now, lower in or less or downregulated or less expressed in these patients was actually interferon programs and cell proliferation programs. Now, these are some of the leading edge genes that were actually driving those programs. And then similarly, we saw that duration of mechanical ventilation was also associated with this sort of gene programs or hallmark programs, where again, patients that had a longer course or were in the longer part, delayed part of their clinical course had an increased expression of TNF signaling program while downregulating some of those interferon programs. And again, this is sort of a representative genes from those programs that are either increasing with time or being downregulated in time. Now, we saw a similar pattern with other sort of non-desirable clinical outcomes in patients with SARS-CoV-2, where you see, again, if you have superinfection, a higher degree of disease severity, again, this program of inflammatory signaling by TNF and kappa B was preponderant. Now, conversely, those patients that had, again, this is our respiratory system compliance, a healthy lung. So if they had a healthier lung, they had increased interferon responses as the dominant gene programs, while downregulating the TNF, NF-kappa B signaling program. Now, this was really interesting to be able to collate gene programs to clinical outcomes. And I think one of the questions that many of us studying viral pneumonia is really trying to understand how are the dynamic changes in viral clearance and the T cell activation programs being changed throughout the course of disease. And so we really know that having an early sort of coordinated innate adaptive immune response really helps in clearing virus and leading to a protective T cell response. And we know that when things don't go that way, then we have seen evidence of immunopathology driven by T cells. And sort of this is an interesting aspect, not only to SARS-CoV-2 biology, but to other virus-induced lung injury. And so in order, again, we have the grainy slides, but the next thing we did was we really wanted to understand this kinetics of viral load and T cell responses. So one of the things we did was we measured viral load by CT value. And so what you can see again here is early versus late samples. So early being here, you see that patients that survived SARS-CoV-2 had sort of a higher CT value compared to those who succumb to SARS-CoV-2, meaning a lower CT value, higher viral load. Now, as disease progressed, this viral load sort of decrease, which makes biological sense. Now, what was interesting here on the right is that you see that sort of decline, or the slope of that decline, was actually more pronounced in patients that survived compared to those that died from SARS-CoV-2. And so the next thing we really wanted to look was how is that correlated with interferon signaling or interferon responses coming from T cells. So this is a pairwise comparison of both CD4 and CD8 T cells, where we look at differential genes expressed that are upregulated in early samples, both here those are the red dots, or the genes that are downregulated here in blue dots. And what you can see, you can't really see, but most of these genes up here are sort of interferon signaling genes, right? And then we did just, for visualization purposes, we did this heat map where we actually structure it or clustered it by timing of mechanical ventilation. So again, this here in dark red and dark green is early mechanical ventilation. As it goes further down, it's late, or so those are late samples. And so what you can see here is that there's increased expression of all of these interferon regulated genes that are early during the SARS-CoV-2 illness. And then correlate here, this sort of purple row is CT values again. So the higher, the lower the CT value, then the higher the viral load. So showing that correlation between interferon signaling coming from T cells and viral load. And so really what this told us was that the early SARS-CoV-2 response was dominated by interferon. Now, we were really interested in trying to know, this was bulk RNA sequencing, so we didn't know really the phenotype of these cells. So we leveraged our prior publication on single-cell RNA sequencing to perform in silico deconvolution of different T cell responses. And what you can see here is that most of these T cells, actually here in the middle panel, are tissue-resident T cells. And there was really no different across different types of pneumonia. The only difference was actually in proliferating CD8 T cells that was seen in SARS-CoV-2. So having really established that these T cells were from the memory phenotype, we really wanted to explore what were they recognizing. So we know that TCR really informs the complexity and the specificity of T cell responses. So we perform bulk TCR sequencing. And here what we did is that we leverage a package known, or an algorithm known as glyph2, which actually, because you get tens of thousands of TCR or CDR3 beta sequences with single chain sequencing from bulk, we are trying to sort of diminish the dimensionality of this into groups that are more likely to recognize a given epitope or a given antigen. So this is what glyph2 does. It sort of parses TCR into specificity groups that are more likely to recognize a given antigen. And so what you can see here is that obviously most of our samples were SARS-CoV-2. So we see a good grouping of this. Each dot here this is a network analysis where each dot is a TCR sequence. And then the connecting gray arrows or lines are the patterns that are shared between them. So showing that there is some sharing between these samples, and that really T cell responses are pathogen specific, as we were able to recognize other known pneumonia causing respiratory pathogens. And so the next thing we did is that as we found these TCR sequences, we were curious to know what parts of the SARS-CoV-2 proteome they were recognizing. I think this has been an interesting question in the field as to are T cells recognizing different parts of the virome, eliciting protective or not protective responses. So this was a question we had. And so what we did is that then we took this abundant TCR sequences and then cross-referenced them with the MIRA dataset from Adaptive Immunoseq, which has 10, I think close to 200,000 high confidence CD8 and CD4 TCRs that have been mapped to a specific epitopes in the SARS-CoV-2 proteome. And we calculated the proportion of this different recognition comparing discharged versus diseased patients. And the thing that we observed here is that discharged patients had a majority of structural proteins typified really by spike and nucleocapsid, while deceased patients had a majority of their samples comprised of ORF1ab complex. Now, again, because T cell responses are dynamic during any response, we wanted to sort of parse this out into early and late samples. And so what we see here is that early on, most of these samples really have an enrichment for this. Again, this is structural proteins here in green and red and orange. But as disease progresses, and particularly in those patients that succumb to SARS-CoV-2 have a preponderance of ORF1ab. Now, again, this another question that we had was how this changes as a function of age, because we know the TCR repertoire changes as we age or throughout the lifespan. And again, what we found was that patients that were less than 65 years of age had, again, a preponderance of more of structural proteins, but those that died from SARS-CoV-2 and were older than 65 had a majority, especially those that succumbed here in the last column, had a majority of epitopes coming from SARS-CoV-2. Now, we next wanted to explore this a little bit further. So we had been able to sort of identify a particular enrichment or distribution of the hierarchy of T cell targets across SARS-CoV-2 proteins, but we wanted to look at specific epitopes. And so this is really, all of this is really computationally inferred, which is, I think it's important to note. But this is, again, a network analysis where each dot with a letter is a given patient. And what we're doing here is we're trying to look at which are the epitopes that are dominant in SARS-CoV-2 response during severe pneumonia. And so every line is an epitope pool or a unique epitope. Most of them are pools. And then the width of the line really represents the magnitude of that response. And so then again, we parse this by outcome. And so what you can see here is that these are given epitopes. If it has an asterisk on the right, it is an epitope pool. And so what you can see here is that the distribution really changes according to outcome. So the dominance and the prevalence of this epitope pools really changes, as well as the breadth of the response. So as in discharged patients, there is sort of a more equal distribution across different proteins. Deceased patients are really dominated by this sort of ORF1ab epitope response. And so this epitope response is really, or T cell recognition of epitopes only, has to occur in a MHC or HLA restricted fashion. So in order to infer HLAs from these patients, we use an algorithm called ARCUS HLA, which computationally infers HLAs in patients. And so what we did here, the top row are it's a given patient. They're separated by outcome. And then the alleles from HLA-A and B, so these are CD8 T cells, are given that the predominant alleles for the 4 predominant alleles for each patient are in the 3rd row. And so then we took the same epitopes here that are the most prevalent, the 5 most prevalent between disease and discharged patients, as well as the most prevalent across a given region of the SARS-CoV-2 proteome, and then try to infer what was the likelihood that T cell in that HLA restricted fashion would recognize a given epitope. And so this is percentile rank or affinity. The lower or the darker the blue is, the higher the affinity binding is, the less dark or light blue it is less of a binding affinity. And so what we found really is that in almost all of these cases, these epitopes, which had been inferred, were also predicted to recognize to be recognized by that HLA, meaning that this renders some kind of validation to our approach. And so the last thing we were interested in was trying to understand the publicity or the publicity of these different TCR responses. So there's been a good amount of work by others showing how the publicity of T cell responses has been associated with outcomes in different scenarios. But really, TCR publicity is how likely it is to find a given TCR sequencing across different individuals. So these are sequences that are preponderant in the population. And the next thing we did here, you can't really see at the bottom, but this is called probability of generation. This is a logarithm 10 base of probability of generation. And probability of generation is the likelihood that a given TCR sequence will be seen or will be formed through BDJ recombination, meaning if a TCR sequence has less insertions or less deletions, it is more likely that this TCR was actually developed or originated during development. If a TCR is, and that means sort of a less negative value, if a TCR has a lower p-gen or a low probability of generation, it is more likely to be part of the post-timic or sort of have been elicited through a specific response, like a virus infection, right? And so we calculated this probability of generation across our cohort and really found that the majority of TCRs were really had high p-gen values. But we did see a significant amount of TCRs, specifically in SARS-CoV-2. Again, these are cross-reference to the MIRA data set, which makes sense, are actually low p-gen probability. And I think what was really exciting is that then we try to parse this out by outcome. And so what we see here is that these patients that actually survive from different types of pneumonia, so this is other viral pneumonia, SARS-CoV-2, and other pneumonia, but not in patients that were control patients, right? So non-pneumonia controls. All of these were actually significant, meaning that the lower the probability of generation of that clonotype, it was more correlated with having a positive outcome in these patients. So this was really interesting to us. And so I'll finalize this part. I want to give 3 slides on some further work, but I think I hope I've been able to convey that T cells that are enriched in the lung space are really good predictors of outcomes in patients with SARS-CoV-2 pneumonia. There are different activation profiles that are elicited throughout the course of SARS-CoV-2 pneumonia that could sort of explain this disparate outcomes in patients, and that alveolar T cells really have also a pretty distinctive hierarchical pattern of recognition of different SARS-CoV-2 proteins throughout the infection process that really could also explain part of this sort of disparate outcomes. Now, I think we all are very interested in trying to understand, why some of our patients, some of them do well, some of them do poorly, but there are some patients that actually linger, right, and don't really fully recover. And we know that, age is really the biggest risk factor for having this sort of bad outcomes. And so I'm really interested in trying to better understand what are sort of the molecular mechanisms that are underlying dysregulated repair during sort of protracted courses in patients that have viral pneumonia and are older. And so I wanted, we learn a lot from RNA sequencing, both bulk and single cell, but it's really hard to give a spatial context to these results. And so in order to try to get a little bit better or a little bit understand better the mechanisms that could perhaps be driving some of these results that we're seeing of this sort of hyperactivated T cells driving poor outcomes, we perform spatial mapping of young and aged mice. We infected these mice with flu, recover their lungs at day 30, and then use a custom panel we made of 300 genes, and then did sort of computational pipeline, and then niche compass, which is a deep graph learning method to infer both spatial niches, and then it also has prior learning, so it allows to understand cellular communication in a spatial context. So we use this to try to infer some of these mechanisms. And so this is the H&E slide from a representative young mouse and old mouse. As you can see, there seems to be some infiltrates here in the young mouse, but they're more preponderant in the old mouse. And so when you look at the youthful response to viral pneumonia, I think it's quite interesting that niche 1 here is actually predominantly composed by sort of the healthy or structural components of the lung, right? With the general capillaries, AT2 cells, and fibroblasts. I think here in red are AT1s. So this is sort of 90% of the lung in this mouse that actually recover from influenza pneumonia. But I think what's interesting here, so this is the old mouse, is that we can actually see that this niche here, which in the young mouse was the healthy one, is almost non-existent. And the preponderance or the majority of the pathological niches that are in these areas of inflammatory infiltrates, you can see that are predominantly composed by these T cells again, CD4 and CD8. We see B cells again. This is cluster or niche 6, which has a sort of a similar composition. And then we have a sort of dysregulated epithelial repair cluster or niche that we find. This niche is also typified by sort of transitional inflammatory endothelial cells and fibroblasts. Now we looked at what are the gene programs that are enriched in these specific niches, right? And just to give you an example and go back to our paper, going to TNF, right? So if we look at the niches that are preponderant for these sort of TLS or tertiary lymphoid structures that have this aggregate T cells and B cells in an organized fashion, we see how some of these signals are coming from this both niche 6 and 0 into our healthy niche, which was non-existent, and also into our airway remodeling niche. So perhaps giving some insights in what are the mechanisms driving dysregulated repair in the lung. So I'd like to end there to give some time for a discussion, 15 minutes for discussion, but this was a huge multi-cross-functional team effort. In Northwestern, we have people across the whole university, the hospital, a really huge effort. But specifically, I'd like to thank Nick Markov, who had the brilliant idea to correlate some of this gene expression profiles with GCA programs. It really changed the course of the paper. CU performed some of the initial pre-processing of sort of next flow pipeline for RNA-seq. Carolina performed deconvolution from our single cell object, and then I sort of supervised this work with Ben Singer. I am a strong proponent of open and reproducible science, so happy to share. Our code is here in the first line. My website, happy to share some of these objects when published, and then oral code is readily available to interrogate and get feedback from you guys. I'd like to thank my lab, Mila, Rat Mila, who performed some of the, Mila performed all of the spatial transcriptomics processing with Rat Mila. And then our RNA sequencing core that are wonderful, and my great collaborator, Lysander. And with that, thank you all, and happy to answer any questions you have.
Thanks so much, Luisa. That was a beautiful talk and a really fantastic paper. I really love the linear visualization of the TCR programs. That's such a cool way of doing that. I'm going to open up and start the panel discussion now with a question for you, Luisa. So if I read and heard you correctly, you said the memory, it was a resident memory T cell response that you were observing at these initial stages, which to me would, based on the timing, and it would kind of necessitate that these T cells would be responding to a cross-reactive, maybe a prior coronavirus response, especially given that, this is a novel virus and, from some of your early samples, they wouldn't have seen it before. So do you think that these resident cells, the ones that are responding with the type 1 interferon, are they cross-reactive? And then how would that change over the course of multiple exposures?
Yes, no, that's a terrific question, Bob, and we went back and forth on this a lot. So most of the patients that came into the ICU usually had at least, I would say, 5 to 7 days of sort of pre-symptoms prior to coming, and getting really, really sick. So there is some lag time there. And now, as I mentioned, this was sort of deconvolution based. So not really single cell phenotyping, and our flow panel was quite limited in terms of surface markers to actually really tell whether these were memory responses. So I think we need, and we're in the process of doing these experiments with employing sort of single cell RNA sequencing to try to really better understand the, in combination, not just pair sequencing of the TCR, but also their phenotype. As to your question about cross-reactivity, that was one that we were really interested in. And I didn't show it here, but we actually looked into the probability of generation of these TCRs and performed sort of a network analysis and trying to see if there was any if there were any sort of low PGen TCRs that were shared between samples from SARS-CoV-2 patients and non-SARS-CoV-2 patients. And we really found just a few of them. The majority were high PGen, so less likely that they were really sort of cross-reactive. But the way the study was designed wasn't really designed for functional analysis of these T cells. So we really don't know whether there is part of heterotypic immunity is actually driving some of these memory responses. So I think future work.
Yeah, absolutely. I mean, it'd be very fascinating to clone a few of the TCRs and see if
they're
stimulated by some other common cold. I have some hands up from Sophie and John, so I'll invite Sophie to ask a question if she has one.
Fantastic work, Luisa, and a great summary for us. I guess getting at Rob's question then, when you're talking about the ability to really draw these T cells in and them being specific through that PGen sort of signature. So the more difficult it is to generate that TCR, the more likely it's specifically being recruited. What are you actually then seeing in like non-specific recruitment? Is there sort of an inflammatory signature where you're basically just drawing in bystander T cells?
Yes, so that's another great it seems, and I guess that's the other part to really tease out in future work, whether some of this, the sort of the circuits with other cell types, because we're seeing some of the signatures, and that's why I show the spatial data. We're seeing signatures coming from neutrophils and IL-6 coming from them. Is this actually also driving bystander activation in some of these T cells that are actually keeping them there? Are these sort of eyeballed or inducible bronchoalveolar lymphoid aggregates that we're seeing in the samples actually part of how are those sort of lymphoid aggregates being sustained in that niche? Where are those signals coming? And I think one very possible one is signals from the dysregulated epithelium, the matrix, and other immune cells that are activating T cells. But really in this study, this was mostly computationally inferred, so hard to tell.
And then to maybe follow up on that, when it's computationally inferred, and it sounds like you really want to revalidate them experimentally, and you've talked about the study limits in the sample collection. Is that due to the sample size from bowel, like the amount of cells that you really recover, or is it due to the ethical kind of constraints of how the study was designed? So what would you like to see in future studies that want to use these samples?
Yeah, so initially when the study was designed, it was not designed to do those functional studies. We sort of, when COVID hit, we kind of adapted some of the collection part of the BAL. But we were pretty overwhelmed, both resource-wise and team-wise, to actually be able to process those samples. So most of the samples were sort of sorted and immediately cryopreserved. So I think future studies, we're trying to modify our protocols to actually in the moment try to analyze and perform functional studies on those T cells as soon as we're able to get them from patients. So it's sort of in the work of modifying our protocols to do those functional assays.
Sounds logistically extremely hard. Are they also able to be frozen? Are they viable long term that you could come back and be like, okay, this is the epitope I was interested in?
Yes. So, after collection of many of the samples, we started cryopreserving for single cell sequencing. These ones initially were not, but they had to be sort of processed immediately after. So, I think a part of what we're trying to do is how can we logistically try to recover, because many investigators have different interests in the sample, so try to have some compromise around the things we'd like to do in the future.
We'll let John jump in here.
Thank you.
Sorry,
muted.
yeah, I'm muted.
Oh.
Great talk overall. This is a fun story. So you mentioned in your cohort you did have some longitudinal samples, right? Do you have longitudinal for the COVID? And did you see a dynamic change in the TCR usage then within a patient?
Yes, I didn't include the figure here, but it's part of one of the figures where we actually in a couple of patients we sort of look at the longitudinal changes in TCR targets in patients that had at least 3 samples throughout time. And so we see sort of that distinctive evolution of initially having more structural proteins and then having more ORF1ab enrichment. So yes, I think a good amount, or because this, as I mentioned initially, these patients really were sick for a long time. So many of them, up to 50%, had superimposed infections as complications throughout their course. So they got repeat sampling. So it's a substantial, not a substantial, but it's a good amount of patients that have longitudinal samples in SARS-CoV-2. In other types of pneumonia, that's not as common. There are some longitudinal samples for that cohort of patients in the other groups, but usually these patients either recover faster or not recover. So the clinical aspect was really peculiar, I would say.
Do you think that's antigen persistence then over time in shifts maybe?
Yeah, so well, that's part of why we did the viral load analysis and majority of those samples actually had lower viral loads. So I guess back to Sophie's point, what is driving, some of the genes that you see there are sort of in the TCR activation pathway, sort of TCR dependent T cell activation. So I guess that goes back to the question, how much of this part of this could be bystander versus TCR mediated. And I think, a different study needs to, a different design or experiments need to be done for that.
Did you have parallel blood samples too to look at antibody?
No, at that time we did not, because really we do it clinically for the patients, right? We try to do the sampling to target antibiotics in them and de-escalate for sort of antibiotic stewardship. But now we are actually doing that. We're collecting, another investigator, Chichi Pickens, is actually collecting blood samples paired with BAL samples to try to see if these signatures match in time. Because the idea is really, can we have sort of a biomarkers, whether they're T cell signatures, a TCR clonotypes that are associated with a good response, a bad response, that can actually help us tailor our therapies when those patients actually hit the ICU, right?
Could I ask a question? I did not figure out how to raise the hand.
Yes, go ahead.
Yeah, so a great talk, Luisa.
Thank you, Jie.
Regarding the NF-κB signaling, I saw the genes you correlate is actually A20 is a negative
Yes.
of NF-κB. So in this case, do you know it's like an over-activated NF-κB or like basically a blunted response or over-activated response?
Yes, awesome question. So part of what I'm trying to look now is sort of, and there's great work from Alex Marson's group looking into CRISPR-based sort of perturbation into TCR dependent T cell activation. And so when he looked at what are the top hits for activators of T cells, right? It is this canonical NF-κB pathway. So you got RELA or P65. You have sort of those, all of the sort of the SAP70 complex, LAT complex. Those are all positive regulators. And as you mentioned, A20 or TNF-alpha-3, right, is a negative regulator. And now the way we interpret this is that perhaps there is hyperactivated, so there's ongoing NF-κB activation, whether that's, because NF-κB also has a non-canonical pathway, which comes through TNFR, sort of TNF receptors, whether that's bystander activation or TCR dependent, that these T cells are hyperactivated, then there's sort of a compensatory increase in those inhibitory pathways. And to your point, and back to sort of the spatial organization of this lymphoid structures, the non-canonical pathway, which it's part of what A20 also inhibits, it is part of it can also be sort of studied under the formation of germinal centers. So a non-canonical NF-κB pathway is necessary for formation of germinal centers, where also these eyeball structures or TLS type of structures are forming with T cells. So we're trying to elucidate whether those are important pathways, which we think they are, because it's part of the signaling that we're seeing in sort of maintaining those inflammatory aggregates close to those areas of dysregulated epithelial repair or airway remodeling, because you see they're sort of in the peripheral part. So yeah, it would be interesting to block A20, well, whether genetically or pharmacologically. Those are sort of the things we're trying to do.
A similar question would be for the IgG response, interferon response. Is this more interferon alpha beta response or interferon gamma response? Or in other words, you getting here would be like autocrine fashion of indifferent gamma response when TCI is engaged during active viral clearance. Does it mean actually the T cell really, the ISG there is kind of T cell really do the job, or a lot of indifferent gamma being produced or not?
Yes. Yes. So to your question, so I think everyone agrees that interferon gamma necessary early on for viral clearance. Now we don't see a lot, at least in this human study. We do see some patients that have lingering interferon expression, at least by bulk RNA sequencing in our T cells, but less so than this sort of TNF activation pathway. We don't see that as much sort of in the latter course of the injury here, and I didn't see it either in our mouse model. So I guess it's something to be explored, but I actually don't know.
Okay, I'm gonna hop in because we are at 4:01. Sophie had a question in the chat, but we might save that for an email communication. And so to wrap things up, I just wanted to thank our panelists, Dr. Valkenburg, Alcorn, and Sun, as well as our presenter, Dr. Morales-Nebreda. Thanks for everybody participating online. And remember, this session was recorded, so if you would like to go back and look at anything or re-access the chat, it will all be up on the ATS website. Thanks so much, everyone.
Thank you.